really excited that you're joining us for this first session. The Rhododendron Research Network is a group of researchers, conservationists, and community members working together to build a global rhododendron community that engages people from all walks of life in the joy and beauty of this amazing plant genus. I'm really excited to introduce our first speaker. As I alluded to earlier, I cited many of Dr. Rajeev Arora's works in my dissertation, and so it is a pleasure for me to introduce Dr. Rajeev Arora from Iowa State University, giving us a talk on ice formation and progression in rhododendron, and a mechanistic hypothesis for winter thermonasty of leaves. And with that, I will say, take it away, Dr. Arora.
Good morning, Juliana. Thank you very much for this very generous introduction. And thank you for putting together this international conference. I'm sure it took a lot of work, and we owe thanks to you. And thank you, everybody, for joining this session this morning. I'll just get right to it because we really have just 15 minutes for our presentations. The main focus of my research in my lab is to understand the physiology of how plants respond and survive freezing temperatures. And over the years, we have sometimes used these beautiful landscape plants, rhododendrons, for our cold hardiness research. And now these broad-leaved evergreens that you see in this picture, they show this very interesting leaf movements, as you can see here under cold, frigid temperatures of winters. The lamina of the leaf rolls like a tube or cigar almost. And also the petiole that you see here in these pictures is also becomes pendant, almost just vertical down. And so together, this rolling or folding of lamina and bending of drooping of the petiole together is called thermonasty, which in other words, it's a temperature-induced nastic movements as opposed to tropic movements. So I find this phenomena quite fascinating, and since it is associated with the overwintering phase of these plants, which is what I am interested in learning about the physiology of cold hardiness, I wanted to learn more about this phenomena, especially in context of actual freezing in the tissue. What's the relationship thermonasty has with actual freezing process? So that's the idea of this presentation. So we conducted this study with this North American species called Rhododendron maximum, very hardy species, native to this area in the eastern United States, in Georgia, and up to all the way to Canada. And this study was conducted with mature plantings of maximum population in this forest. You can see all these details here, a little bit about the map where this site is located. And these are mature plantings. And so we wanted to visualize, the goal was to visualize the freeze initiation in real time, ice progression once the ice has formed, how it moves inside the plant, and also the thermonasty under natural freeze events. And there were 4 independent freezing events on these dates, as you can see here in November and December and January. That's when this experiment was conducted. And what we utilized was infrared camera to actually see these things real time. So we did infrared videography on 4 freeze events. This is some of those pictures to show you the site. This was cleared up. This forest was cleared up, 2 meter by 2 meter area where you had several plantings we used. And within a plant, we used several branches. So it was fairly well replicated study. Here's the picture of my collaborator, without whom this work would not have been possible, because he is the person who had the cameras and had the expertise to do the infrared videography, David Livingston. He has now retired from USDA ARS, but he is a plant physiologist, and he was at NC State campus. So the 2 questions that we really asked in this study was, number 1, what is the temporal relationship between freezing and thermonasty? In other words, which precedes the other? Because in literature, there have been some confusion about when this actual rolling of the lamina occurs. In some in some reports, it suggests that it happens even before freezing. So we wanted to actually test that by this real-time freezing technique. And also, by doing all that, can we learn anything about some putative mechanism of thermonasty? So these are the two questions that we asked. Just a quick setup of this study. Essentially, these are the video cameras that were mounted on these stands. And the monitoring of this freezing process began at about 8 to 10 p.m. at night, when the temperature was not quite below freezing at that time. And it ended in the morning when the temperature dropped down to as low as minus 9 degrees Celsius. In fact, one night there was temperature about right around minus 10 degrees Celsius. Okay, so now in this slide, there's a video that I'm going to play for you. That will go very fast because it has been sped up so that you can see the entire thing in just a few seconds, although it took several hours for this whole process to be completed. So on the left is the video, which I'll play shortly here. And on the right, you have several still photographs that were extracted from this video to show you the sequence of this progression of this whole process. Okay. So the freezing begins at about -2 to -3 degrees Celsius. And this is very similar to what we have seen before also, and others have reported that typically in nature, when you have all the nucleators, the freezing initiates right around -2 to -3 degrees Celsius. So not much supercooling. One thing you should note is that the color will change as the ice will form, because when the ice forms, it gives off heat. So this is an exothermic event. And the color of these leaves will go from quite dark right now to a little bit grayish and light in hue. Okay, so that's what will tell you the ice is formed and also how it progresses. Okay, so let me play this video. This ice is forming right now. You can see it's moving into these veins. You will see now this one is moved right now. So, and then after ice has formed, this drooping of petiole and the curling of lamina occurs. So ice forms, like I said, about -1, -2 to -3 degrees Celsius. Shortly thereafter, petiole drooping intensifies, and the leaf also becomes completely downward, or vertical downward. And the maximum thermonasty positioning occurred almost 2 to 3 hours later by the temperature. At that time, the temperature was about -5 to -6 degrees Celsius. As I said, the similar response was observed in all those freezing events and in all the replicated examples. And of course, next morning when the warming happened, the leaves will unroll and the petiole will go right back up into its more ambient kind of position. So the main point of this slide is that there is no additional exotherm observed anywhere else other than the vascular system. Okay, right here, which is the midrib here and the side veins. That's where the ice forms, and no additional exotherm was observed anywhere else. So this basically answered our first question, that the freezing precedes laminar rolling and intensified petiole drooping. I say intensified petiole drooping because some petiole drooping is already present when even the freezing has not occurred. So, but it becomes much more intensified after the freezing process. Okay. Okay, so we already saw that freezing initiates in the stem, moves into the leaf via its mid vein and goes into the side vein, and then only the rolling and the drooping of petiole occurs. But we wanted to further check and confirm that the part of the leaf that freezes is indeed vascular system. So we wanted to see the progression of ice actually inside the petiole and leaf. And that actually is shown by the help of these two videos here. So there's a video on the left and another video on the right. And again, these would be the still images extracted from these respective videos here and here. Okay, here is a histology of petiole, just internal structure here. Okay, so let me just explain that what the setup here is. So this is a petiole which is attached to the stem, and this petiole has been cut longitudinally. Okay, so you can see this vascular trace almost here. So we are actually looking at inside of the petiole because it has been cut longitudinally. The lamina has been cut away from this petiole, but petiole is still attached to the stem from where the ice actually is progressing into the petiole. Okay. Here you have an actual leaf, which is also attached to the petiole and to the stem. So this is all happening in real time. And this leaf has been cut in a transverse section. So again, here you are seeing inside the leaf. You can see the vascular cylinder here of the mid vein, and there'll be several vascular bundles here of the minor veins right here, as you can see in these pictures a little bit here. And we pictured these things with the help of a microscopic lens, which was mounted onto this infrared camera. So the camera is mounted with the lens, and the lens is facing these cut surfaces, as you can see. And that's when we started the videography during this freezing event. Okay, so let me play this for you. So you can see this ice has formed, goes into this vascular cylinder here. As you can see, the color change due to exothermic event. And now it will get sped up to show you the whole thing in just a few seconds. And once the ice has formed, and then you will see very quickly this petiole drooping here. Now let me play this video. Here you will see all these things popping up like a firework. Here the ice has formed, and it will go into this other side veins. So all this change in the color is because of this exothermic event. Okay, and so this basically is further confirming that ice forms only in the vascular part. So leaf rolling initiated and petiole drooping intensified after freezing in the vascular system, and no freezing was observed either in epidermis or mesophyll tissue. It's only in the vascular system. Okay. So here is our hypothesis based on what we observed. We believe the rolling part, at least, lamina rolling, occurs due to this differential contraction of adaxial versus abaxial side of the leaf due to freeze desiccation. So let me just explain adaxial versus abaxial surface for those who might not know these terms. So here is a leaf anatomy, essentially, of these rhododendron maximum leaves. This is a cuticle upper here, and then you have an epidermis, then you have palisade, and then you have spongy parenchyma. These are the vascular bundles. So the upper side of the leaf is adaxial, and the lower side is abaxial surface. Okay, so when the freezing happens, the ice at a given temperature always has a lower vapor pressure than the supercooled water. Okay, so when the ice forms outside a cell, it will pull the water out because of this vapor pressure gradient and will cause the cells to contract, like is shown in this drawing here. So we believe that the lamina rolling happens because these adaxial versus abaxial cells of these leaf are contracting differentially. Some contract more than the other. The idea is basically what we are suggesting is that those cells like spongy parenchyma, which are on this abaxial side, which are very, very close to the vascular bundles, and those palisade cells that are also proximal to the vascular bundles, they will dehydrate first, and epidermis, upper epidermis, and the palisade that are further away from the vascular bundles will dehydrate and contract later and less. And that will kind of, with the help of this thick cuticle, which we know that rhododendron leaves have very, very thick cuticle. And in fact, in the winter time, this cuticle deposition becomes even more intense. And we have done a lot of work, and I don't have time to show you the pictures, but we have the data to show that. So this thick cuticle will actually reinforce that differential contraction by these two sides to cause this leaf rolling. So that's our hypothesis. Now, to expand on that working hypothesis a little bit more now, what we are suggesting actually is that this rolling occurs because of this anisotropic change in the shape of the cells in adaxial and versus abaxial surface in response to freeze desiccation. So freeze desiccation is the key, of course, that is drawing this moisture out of those cells. But these cells from adaxial versus abaxial, they are contracting anisotropically. Now, anisotropy is a concept that we used in that is used in physics, essentially, quite commonly implicated in material science and wood science area. And very simple example that I can think of for to explain anisotropy is warping of wooden planks or doors in your house. When you have a lot of humidity around, the doors warp a little bit, the wood warps, and that is happening because some cells are expanding by absorbing this moisture faster and in a different direction than those other cells that are contracting less and in a different direction. And that whole thing is called anisotropy. Okay, the physical mechanical property of material is different when measured along different axes. So what we are suggesting here in the context of this leaf rolling, instead of expansion, of course, here things are contracting because these leaves, these cells are losing water. So in the context of this leaf rolling, we believe it is differential contraction of adaxial versus abaxial sites, or anisotropic contraction. And I try to sort of draw by my hand to give you a little bit further sort of visual on what I'm thinking is happening. So here you have a palisade. This E is missing here, sorry. And here you have spongy parenchyma. So this palisade, the vertical sort of file of these cells right here, they are contracting a little bit less and also more horizontally, whereas the spongy parenchyma cells, which are here more close to the vascular bundles, they are dehydrating earlier and more, and also more into this kind of orientation. And a lot of this anisotropic behavior of these cells is most likely governed by or influenced by the cell walls. We know that cell wall is the most rigid part of the cell, and so and you have this very complex structure where this cellulose microfibrils are traversed into this matrix of pectin and hemicellulose and so forth. So how these cellulose microfibrils are oriented and what's the architecture will determine how the water intercalates through these cellulose microfibrils. And when the water is lost during this freeze desiccation, it might be very different from different cells depending upon the cell wall structure. And of course, the midvein does not roll. That works like a brace. So reinforced by thick cuticle, this rolling will occur. So that's our actual working hypothesis. Now we wanted to test this hypothesis a little bit further with a very kind of a crude looking model here, which is a cellulose-based paper-bilayer model. So the only resemblance between this model and the leaf would be just the cellulose part, which is, of course, the cell walls are made up of cellulose. Otherwise, there is no other similarity. So what we did was we had these two paper sheets that were cut in identical length from just a basic tablet, paper tablet. And it was cut in two directions, one against the lines and one with the lines. So these are the lines right here on this tablet, on this sheet. So one along the lines, one against the lines. And we put those two sheets together. They were moistened. And when we moistened them, we found that the paper that was cut against the lines expanded about 2.4% in length upon wetting, whereas the one that was cut along lines did not expand. So you can already see this anisotropic behavior here. Okay, and they were joined by this silicone caulk, these two sheets, after they were wetted. And when you dry them, the shrinkage in the expanded layer was resisted by the other partner, which did not expand, and that caused these paper sheets to warp like that. So this is what we are trying to sort of convey here, that this anisotropic expansion by moisture absorbance by these two sheets is causing this to roll like that. When you place it back in water, this bilayer becomes straight again, and that was not shown here. When you freeze these bilayer, just like when you dry them, this also behaves the same way. So this is a very simple bilayer cellulose-based model that behave anisotropically and responds very, very similar to how the leaves respond when they roll during freezing. And here, just to show you some of those structure of cellulose, and it has a lot of hydroxyl groups, and have a lot of affinity for water. It attracts water molecules. So as I said, depending upon how these cellulose microfibrils are oriented in these pectin matrix, and how the water molecules intercalate will cause this anisotropic contraction in the case of leaf rolling. This is my last slide. So here, the answer to number 2 question, where we are trying to learn a little bit about the mechanism of thermonasty, we believe that it is a combination of 2 processes. It's a combination of biological process as well as mechanical process that explains this leaf rolling, at least. The biological part comes from this efflux of water from the cells due to this vapor pressure gradient, and that efflux must take place through membranes that surround the cells, and those membranes have these water channel proteins which are called aquaporins. So it is through aquaporins of this basophil and epidermal cells that the water goes to the vascular system during freeze desiccation. And here is a slide, couple slides here from our earlier paper that we published in several years ago, which was conducted with another North American species, Rhododendron catabians, also very, very hardy species. And this experiment was done in controlled conditions in a growth chamber, where we exposed these potted plants as well as excised leaves to different temperatures to look at the freezing, and extracted these RNA from these leaves to do the gene expression studies and so forth. And so we found was that the ice nucleation, the actual freezing initiation, happens right around -2 degrees Celsius. And it is also coincident with that. You can see these 2 aquaporins called RC-PIP1 and RC-PIP2, Rhododendron catabians PIP1 and PIP2, also show this higher abundance of expression in these leaves. So this is kind of a indirect evidence. Of course, we do not have an idea about the activity of these proteins, but we believe that these might be involved in this water efflux. And the mechanical part comes from the fact that we found that when we have these maximum leaves completely dead by putting them in oven overnight, they also roll just like a live leaf would. So this is purely a mechanical process also, this, and again, due to anisotropy. Some people call this as hygronasty, because this is happening due to just desiccation, pure desiccation, not freeze desiccation. So this is our hypothesis, and that pretty much concludes what I had to present today. I hope I didn't really go over time. I was not even looking at the clock. Thank you all very much. If you have any questions, I'll be happy to answer the questions.
I think we have plenty of time for questions. So the ways to do that is you can raise your hand, you can put a question in the Q&A, or you can put a question in the chat. So feel free to do one of those 3 modes right now to ask a question. So if there are no other questions, I have a question for you. So one of the things that I've been working on with a collaborator is a really neat data set showing environmental temperatures during leaf thermonastic events. And we have, there's a film of these events. So what would you suggest we look for in our film?
So what species you are working with?
It is Brachycarpum fowleri, so closely related to maximum and catabians, I think.
So you are saying that this film is this is just a film. They're just showing you during freezing events and thermonasty and stuff like that.
And we have the temperature and we have the film.
So how do you get the temperature?
From the camera, it records the temperature.
So it's an infrared camera?
No, it's just a video.
Just a video. And that will show you the temperature, huh?
The camera has like a temperature sensor, so.
Okay. So yeah, that the thing is with this infrared videography. So basically, what from that film you would be able to at least see whatever temperature monitoring method that camera has, whichever way it is actually recording the temperature, that will be the air temperature, perhaps, and not necessarily the tissue temperature. And also, we have seen in our experience that when we used to use, before this camera, we used to use, and people still use, and I still use it too, is what is called thermocouple, to actually measure the temperature of the tissue, if you want to really get very, very close to what's happening. But even thermocouple is only giving you a temperature of that particular spot where thermocouple is inserted.
Right.
And the ice formation might not be happening at that point, because you can see from my pictures here, at least in rhododendron maximum species, the ice forms somewhere in the stem, and then from stem it goes into the vascular system of the leaf through the petiole and so forth, and that's when the thermonasty, or this rolling and drooping, happens. So that temperature, first of all, from this movie, if you let's say happen to find for this species that the temperature is, let's say, minus 3 degrees Celsius, uh, and uh, and thermonasty has not even occurred yet, that is not necessarily disproving the fact that no ice has formed, and yet the thermonasty has not happening, because we really do not know if indeed the ice has actually gone into the leaves or not. We do not really know unless we really know some kind of an exothermic event where it's coming from, and we can, in this case, of course, we can visualize, but you can have a bunch of thermocouples attached to the leaf where you can see, and that thermocouple has to be attached to some kind of a monitoring device to see that, okay, there is a, in fact, release of heat, ice has formed, and then you can start to correlate with the rolling or drooping of petiole. So
Yeah.
yeah, this movie will maybe give you some kind of interesting information, but it might not give you a very high resolution understanding of actual freezing process in relation to thermonasty, so that you can really draw some actual conclusions that, okay, indeed, that's when the freezing happened, and that's when thermonasty happened.
Yeah, it's very complex.
It is very complex.
I'm glad I saw your video because now we'll be more careful interpreting our data. We have time for one more question, maybe from the audience. Jean Burns asked, any speculation about relationship between freezing and drought response?
Um, Freezing and drought response. So, of course, these kind of movements you see, leaf movements you see in rhododendron during very, very dry conditions also. And as I said, in this whole process, so of course the commonality is loss of water in both of these phenomena, freezing or dehydration due to drought. Let's say it's the common feature here is the moisture loss from the cells. So that contraction in what we believe is anisotropic contraction is happening in both cases. Now, in the case of drought, when it's a pure lack of water, that is out of the two possibilities that I explained in my last slide, that biological and more mechanical part. So the mechanical part becomes more predominant in the drought case, because there you're just simply drying. There is no water there, essentially a very, very low water potential. Whereas in the case of freeze desiccation, um, it's actually water is not losing the plant. Water is only going from cells, mesophyll cells, to the vascular bundles where the ice is. So water is still present inside the leaf. The overall water balance is the same, but cells are dehydrating because the water has moved to the ice locus in the vascular bundles. In the drought, actually it is the loss, or entire tissue is desiccating. So that's a difference. Otherwise, desiccation or loss of water is common in both. In one case, it is losing the plant, or in other case, it's just losing the cells and moving to the ice front.
Oops, I'm muted. Okay, we have a couple more questions, but I think we want to move on to our next speaker, Rajiv. You
Okay.
can answer those in the Q&A.
Okay.
So I'm gonna send you, so let's try to get those answered in the Q&A if you can. Can you see those questions that are still there?
Yeah, actually, Michael Bamford, in what form does
Yeah.
the body replicate the cell, and where does the molecule expand once frozen?
So we're gonna, if you want
I'll
to
go ahead.
answer that in the
I'll
chat,
type that. Yes, I will do
sweet.
that.
Or in the Q&A. So what we're gonna do is we're gonna move on to our next talk.
Okay.